On a dead world, one suitcase of living Earth soil could be worth more than a vault filled with diamonds? At first, that sounds ridiculous. Diamonds are rare, guarded, polished, and priced like treasure. Soil is the stuff we wipe off our shoes. But picture us inside a settlement on Mars, hungry, cut off from Earth, and trying to grow the next harvest. The diamond still sparkles. It just cannot feed anyone. Our first plan would probably seem obvious. Scoop up the local ground, add water and fertilizer, plant seeds, and call it farmland. Brown or gray powder looks close enough to dirt. How different could it be? Very different. The question is not whether another world has crushed rock. Many worlds do. The real question is whether productive soil is simply a material we can find anywhere, or a living planetary system we would have to rebuild piece by piece. Start with a transparent jar of sterile crushed rock. It contains minerals. That matters. But it is only the beginning. Crushed bricks are not a house, and loose mineral particles are not a completed soil system. In 1941, soil scientist Hans Jenny formalized an influential way to understand why the starting material matters. But so do climate, organisms, landscape, and time. Put similar rock in two different places, and the outcomes can diverge because water moves differently. Temperatures change at different rates, slopes drain or erode, organisms arrive, and time allows layers and structure to develop. Rock is the raw dough. Climate, life, landscape, and time supply the mixing, fermentation, heat, and baking. Grinding the rock more finely reproduces the flour, not the finished loaf. Now add water to our jar. The particles darken. Temperature changes crack minerals. Dissolved compounds move downward. Some materials are removed, others accumulate, and recognizable layers can begin to form. Tiny spaces appear between particles. Some hold water, others hold air. The material is no longer just sitting there. It is being transformed. That is why two neighboring patches of ground can behave differently, even when they began with similar rock. Soil carries a history. It records what happened to the material, what moved through it, what lived in it, and how long those processes continued. Earth had a very long head start. Our planet formed about 4.54 billion years ago, but the soils we recognize today were not present from the beginning. The planet supplied stone long before it completed anything resembling a garden. Physical and chemical weathering worked on exposed material. Microbial communities occupied land before forests existed. Then, roughly 470 million years ago, early land plants became established in landscapes already inhabited by microorganisms, algae, lichens, and fungi. Life did not simply move onto the land. It began changing how the land worked. Roots entered cracks, fungal threads spread between particles, microbes transformed organic material, dead matter accumulated and decomposed. Soil animals moved through the ground. Plant and microbe-derived compounds help particles bind into aggregates, while roots and fungal hyphae physically held some of those particles together. Return to the jar. Organic fragments are now mixed with minerals. Roots divide the material. Fungal threads cross the gaps. Microbes gather around them. Pores hold both water and air. What looked like powder begins behaving more like a habitat. A handful of healthy soil is less like a bag of dust and more like a crowded underground city. Minerals are the buildings, but roots, microbes, fungi, water channels, air spaces, and decomposers keep the city operating. One recent estimate suggests that soil is home to roughly 59% of Earth's species. Not exactly empty dirt. Those organisms help influence decomposition, nutrient cycling, water movement, aggregation, and other major functions. Remove the community and the material may still look similar, but recycling slows. Structure weakens, nutrients may remain unavailable, and the hidden city stops doing much of its work. That brings us to the moon. Place a second transparent jar beside the first. Fill it with lunar regolith. At a glance, it may look like another kind of dry soil, but its history is completely different. The moon's surface has been shaped largely by repeated impacts, fragmentation, melting, and exposure to solar particles. Its regolith contains broken rock, mineral grains, glass, agglutinates, and fine dust. It is the product of a cosmic rock crusher, not Earth's rain. Roots, decay, flowing surface water, and known soil ecosystems. The moon gives us abundant mineral material. It does not give us ready-made farmland. The particles can be sharp and reactive. There is no naturally developed organic layer, no confirmed biological community, and no established nutrient cycle waiting for a seed. Our simple plan, add water, add fertilizer, grow dinner, has already lost several invisible pieces. Still, the story does not end there. In 2022, researchers planted Arabidopsis thaliana in genuine regolith returned by Apollo 11, 12, and 17. The seeds germinated. That matters. Real moon material did not automatically prevent a plant from starting to grow. For a moment, the obvious plan seems rescued. Perhaps regolith does not need to become Earth soil. Perhaps plants only need the right assistance. Then, the rest of the experiment appears. The plants developed more slowly, remained smaller, showed visible stress, and activated genes associated with salt, metal, and oxidative stress. Water, nutrients, air, temperature, and light were supplied under controlled Earth conditions. The regolith was part of the growth environment, but it was not running the system. A seed can open inside a damaged building if we carry in food, water, heat, air, and electricity that does not make the building a self-sufficient city. This changes the mission. We are no longer searching for the correct kind of dirt. We are asking whether we can rebuild a planetary process. Soil is not merely the substance beneath an ecosystem. It is also one of the products of that ecosystem. Mars seems like the next more promising candidate. Its surface material has interacted with wind, ancient water, ice, volcanic minerals, radiation, and atmospheric chemistry compared with lunar regolith. It offers a more varied geological history. Maybe Mars has already completed more of the work. But in 2008, NASA's Phoenix lander detected perchlorate in material at its landing site, at approximately 0.4 to 0.6% by mass. Perchlorate does not prove Mars is lifeless, and it does not mean every location has the same concentration. It does create an important chemical complication for crops and human use. The construction material arrives with a warning label. Mars is also thin-aired, cold and dry in its present environment. No living Martian soil ecosystem has been confirmed, so even where the mineral material is useful, we still need treatment, water management, nutrient balancing, organic inputs, biological partners, and protection inside a controlled habitat. At this point, our instinct changes. We stop asking whether alien ground is soil and start asking how many soil functions we can manufacture. A realistic engineering path exists. We can adjust particle mixtures to improve drainage and aeration. We can treat problematic chemistry. We can supply nutrients and make mineral nutrients more accessible. We can add composted waste or other organic matter. We can introduce selected bacteria and fungi. Inside a greenhouse, We can control water temperature, pressure, gases, and light. Recent experiments with regolith simulants make that possibility more than fantasy. Biological and material amendments can improve plant performance under controlled conditions. In a 2026 study, mycorrhizal fungi helped chickpeas flower and set seed in mixtures containing up to 75% lunar regolith simulant and vermicompost. But every plant grown in 100% simulant died before completing the experiment. That is real progress and a real limit. We can begin hiring replacement workers for the underground city. Fungi can help roots. Microbes can make some nutrients more available. Organic amendments can improve the material. Yet those organisms also need suitable water, temperature, chemistry, energy sources, and continuing organic inputs. Every solution introduces another system that must remain alive. The jar of lunar regolith now has tubes entering it. One supplies water, another supplies nutrients, a chamber maintains pressure, lamps provide light, heat keeps the roots within a usable range, organisms are added, the jar is becoming productive. But the support machinery around it is growing faster than the plant. Then comes the harder test. Not one seed, but repeated harvests. A colony does not need a plant to germinate once it needs crops to grow through many cycles. Nutrients removed in food must return. Waste must be processed without allowing toxins, pathogens, or salts to accumulate. Microbial populations must remain balanced. Soil structure must not collapse. Water must keep moving through the root zone. Roots must receive both moisture and air. On Earth, the surrounding planet performs much of this environmental work. Away from Earth, machines may have to perform it first. That creates the reversal. Turning regolith into a productive growth medium may be possible if we provide safe chemistry, accessible nutrients, suitable structure, organic carbon, selected organisms, atmospheric control, temperature control, waste recycling, energy, and continuous monitoring. But supplying all of that creates dependence on imported biology. Treatment equipment, habitat machinery, replacement parts, and long-term ecological management. The more soil-like functions we add, the more of Earth's living system we discover we are trying to rebuild. So we must separate three very different achievements. A laboratory growth medium can help a plant survive under tightly controlled conditions. An engineered agricultural substrate can support useful crop production with treatment, amendments, and machinery. A natural resilient soil is a developed system that stores and moves water, cycles nutrients, supports biological communities, maintains structure, and continues functioning through changing conditions. Those are not the same thing. Earth remains the only world where life and therefore a living soil ecosystem has been confirmed. That does not prove Earth's soil is unique in the universe. It means it is the only confirmed example we currently have. Even here, soil is slow built. FAO estimates that about 95% of human food depends on soil, while only a few centimeters of new soil can require centuries and in some settings up to 1,000 years to develop. A layer thinner than a finger may represent many human lifetimes of accumulated work. Now return to the Dead World marketplace. The vault is open, the diamonds are flawless. Beside them sits one dirty suitcase. But the suitcase no longer contains mere brown material. It contains weathered minerals, organic carbon, water-holding structure, living communities, nutrient cycles, pores filled with air and water, and part of Earth's biological history in miniature. It resembles an operating planet. The diamond has not changed at all. We may eventually use lunar or Martian regolith to build productive agricultural systems. We may treat it, amend it, inoculate it, and place it inside habitats where machines keep the necessary conditions stable, that is scientifically plausible and increasingly testable. But we would not be discovering ready-made Earth soil. We would be reconstructing the relationships that make soil function. So, on an isolated world, living Earth soil really could be more valuable than diamonds. Not because science has assigned it a universal price, and not because it has been proven the rarest material in the cosmos, but because value changes when survival is the market. A diamond can preserve wealth from the old world. Living soil can help create a new one.